Here we go. And our speaker today is Dr. Fabienne Rössler from ETH Zurich in Switzerland. Fabienne completed her undergraduate and graduate studies at the University of Zurich, where she began with a focus on neuroscience, but then transitioned to data science and machine learning in the biomedical field. After that, she pursued her PhD at the Norwegian University of Science and Technology, where she was specializing in data science approaches for analyzing clinical and molecular data of lung disease patients. Then she moved back to Zurich and currently conducts her postdoctoral research at ETH Zurich, where she applies data science approaches to basic biomedical research. And specifically, she develops new approaches for deep behavioral phenotyping in rodent preclinical models in the field of stress and neuroscience. And the title of the talk, Using Behavior Flow Analysis, BFA, to uncover latent behavioral phenotypes, the recently developed software tool. And we welcome you, Fabienne. Thank you for your time. And now floor is yours. Please.
Thank you, Vootele, for the very nice introduction and also the opportunity actually to present here today at the TEATIME webinar. Yeah, as you already said, today I want to talk about the behavior flow analysis, or shortly BFA tool that we recently published and that we propose to uncover latent behavioral phenotypes. So today I will talk a lot about behavior, and as most likely many or most likely all of you know, that, let's say, back in the days, behavior analysis was mostly done manually. So a human had to, I don't know, watch the behavior test itself, or maybe also offline in video recordings, and monitor specific behavior. They were interested, for example, I don't know, it could be rearing or grooming, head twitch, whatsoever, in these behavioral setups. However, I would say now in the last 10 to 20 years, there are certain tools popping up all over the place, let's say, that, well, some of them are related to machine learning, AI, but of course also other tools that try to automate this very laborious tasks, and to kind of, well, the hope is to give us more time for other tasks instead of just watching these videos and monitoring all this behavior, etc. So when I talk about these tools, I want to mention one specific, and this is this marker-free pose estimation. And I just want to show a few examples from our own lab, where we have, for example, here in the upper left corner, we have an open field test, we have a light-dark box, we have a marble bearing test, and, for example, also an elevated plus maze. And as you can see now with this pose estimation tools, we are able to track the mouse in all these setups quite reliably. Well, not the whole mouse, but specific points that we defined previously. So we kind of know all the time where the mouse is. We also normally track actually the arena it's placed in. So, for example, here the corners of the open field test, but also of the other tests. And this gives us, or in general gives people the opportunity to, for example, kind of automatically get some of these classical readouts. For example, in an open field test could be distance moved, time and center. So to get all this information out of the recordings automatically. But of course, you can also go to, I would call them a bit more advanced analysis methods that are based mostly on these pose estimation tools. And I want to talk about, let's say, two specific roads that you can go when you do behavior analysis, or when you try to automate these behavior analysis to a certain degree, and that's the supervised versus the unsupervised behavioral analysis. And I want to start with the supervised analysis. And what you normally start with is, well, with your experiment, and you record your mice. And what then happens is, if you want to train a model to detect specific behaviors, for example, rearing, grooming, walking, running, jumping, whatsoever, you need a human that first of all designs this set or decides about these sets of behavior you want to detect, and then labels training data for the model. So one or normally several humans kind of look through a set of these test videos and say like, okay, that's a rear, that's a groom, and so on. And this training data is then later on used by most cases, a machine learning model that uses this training data and then tries on features that are computed based on these tracking points. That can, for example, be speed, acceleration, distance between certain points, tries to learn how to recognize these defined behaviors. And when it's done so, it can be used on new data to classify these kind of behaviors automatically, and you can then just run your statistics and see if you find effects or differences between, for example, two treatment groups. Now, if you have a good model and it does a good job, you can get quite a high accuracy in detecting known patterns and behavior, and this in an automated way. So you don't have to do the work yourself, you can just let the model work for you. However, there are, of course, also some limitations to this kind of approach. It, first of all, needs labeled data, which is quite laborious. Well, it depends a bit on what kind of behavior setup or kind of behavior test you're interested in, and maybe also the behaviors you want to look into. But at least to my own experience, this takes normally quite some time and human power to kind of get these labeled data. On the other hand, another limitation is that by defining this set of behavior, you may miss novel or hidden phenotypes or hidden behavior in your behavioral setup or behavioral tests. Now, to kind of address these limitations, we have now also the unsupervised behavioral analysis pipeline, let's so call it, where we normally also start with an experiment. You record your mice, and what happens then is you take this data and you just give it to again, a model that based on, again, these features that you compute on these tracking points, tries to kind of find pattern, segmentations, clusters that resemble, hopefully, some meaningful behavior. So what you normally get is kind of these clusters, and what you can then do, and this again needs to be done by a human, you can look at these single clusters and kind of relate them back to known behavior that we can also interpret. For example, I don't know, this could be cluster 2 that you then say, okay, that's a rearing cluster for me, or cluster 7 that could be a grooming cluster. And of course, on that, well, you actually don't even need this kind of association, but you can then also run your statistics on that and see if you find differences between these groups. There are nowadays several tools available that have been published over the last 5 to 10 years. I just want to show you a few. For example, BSOID, VAME, MOSEC, Keypoint MOSEC is the most recent one. So for that, you don't even need to come up with your own solution. You can just use these tools, at least in principle, you can use them, apply them to your data and see what kind of clustering or results you can get out of this. Now, we were also using this kind of pipeline with the unsupervised approach. However, we faced certain limitations. And I now don't want to talk about, of course, all these methods have certain limitations. They compute different features, which can then lead to different outcomes. They make certain assumptions about how your data needs to look like to find nice clusters in the data. However, kind of what we in our work want to address is even later on. So we're not proposing any new behavior segmentation tool. We're actually looking at the statistics that you run on these outputs. And we see a few limitations there that we wanted to address. And the first one, and this is now just a bit of an overview, what I want to talk about in the next half an hour, roughly, to give you a bit of an overview, what you can expect. So the first limitation that we normally faced was the issue with the multiple testing correction. And we kind of try to address this with the so-called behavior flow analysis, or BFA. Then there was also a bit of the issue normally that we wanted to compare individual behavior profiles, maybe also see how do single animals respond differently to a treatment. And we also wanted to come up with a tool that makes this comparison actually a little bit easier. And for that, we developed the behavior flow likeness, or BFL score. And then at last, we also wanted to have a tool or kind of a pipeline, maybe more, that makes it easy to actually compare behavior profiles or behavior phenotypes between experiments. So not only inside of one experiment, but maybe even between experiments. And for that, we kind of came up with what we call the behavior flow fingerprinting, or BFA. Now, before I kind of dive into these different topics and explain a little bit more what we actually did and how you can use them, I just want to make 2 disclaimers. So we are Stress Lab. We mostly work with mice. So all the data that I will show you later on is based on mice and mice behavior data. However, all these tools in principle can be used on whatever behavior tests and whatever animal models you want to use. So if you do experiments with rats, or if you have zebrafish as an animal model, of course, all these tools would also work, as long as you kind of can track the animal and can do some sort of clustering or behavior segmentation on this data. And then the second disclaimer would be all the data that I show you now with the clustering is based, so we normally use a k-means clustering in our case. However, as I showed you before, there are other segmentation tools available, and they work also perfectly well with this whole pipeline. So as I said, we are normally more addressing the kind of statistics afterwards. So if you already have a running pipeline with, for example, VAME or MOSEC, you could in principle still use all these tools here. So yeah, okay, good. Then now let's go to the first point I want to address, the issue with the multiple testing correction. And here I shortly want to show you a bit what I mean. So when you cluster your behavior, what you normally get, you get, well, you normally choose the number of clusters in many cases. So for example, in this case, we chose 25 clusters, and you can then just count, for example, the occurrences of these clusters in the different groups. Here we have 2 groups, control and side. And then you can just run your statistics. So you run, okay, you say like, okay, how does this compare for the different tests? And in this case, what you end up, so the number of statistical tests you have to run is the number of clusters you choose to analyze your data with. Another way to look at your data is to look at cluster transitions. And that's also what we normally go for now. So most of the data I will show you is now based on the cluster transitions, because here you kind of preserve the temporal information in your behavior data a little bit more than what you have here. So what are cluster transitions, or what do we mean with cluster transitions, is that, for example, if we have behavior 41 or cluster 41, how many times does it transition, for example, into cluster 23? Or how many times does cluster 23 transition into 69 or 45? This can then be, for example, visualized in such a circular plot. And of course, you can also quantify these transitions and then run statistical tests on that and see, okay, are there certain differences between the 2 treatment groups we have? And here, we even get more statistical tests we would need to run if you really want to assess all of these transitions. Here it's the case that we normally have number of clusters times number of clusters minus 1. For this specific case where we chose 25 clusters, that would mean that up here we have 25 tests and down here we have 600. And that means that we would need to apply multiple testing correction. Because if you run so many tests at the same time, we just have a high chance that we will detect a lot of false positives. And to reduce this chance, we should apply multiple testing correction. Previously, if we only have classical readouts, which are normally just 1 or 2 maybe, for example, in open field tests, if you only assess distance moved or time in center, you don't have to worry about this. But now, because we're dealing with so much information in the end, we have to think about this. And this can become an issue, especially if we have very high numbers of, for example, in the case of transitions, that this can be quite punishing. So you lose a lot of your, let's say, significant findings due to multiple testing correction. And to kind of address this issue a bit, we came up with the behavior flow analysis, or shortly BFA. And I want to show you a little bit what the idea behind it is. I will not go into too much into like mathematical details, but just to hopefully get you a bit of a feeling what we're trying to do, so you can also better then interpret the data I will show you on later. So, as I said, we are normally working with cluster transitions, and how this looks like is what you normally get is such a transition matrix. So you have maybe cluster 1, 2, 3, 4, also up here. And what you see here, for example, for this specific animal, cluster 1 transitioned 11 times into cluster 2, or cluster 4 transitioned 7 times into cluster 1, and so on. So that's kind of how your data looks like. And you can, of course, get this information for several different animals, your control animals, and your test animals. Just made some examples up here. And what we want to do now, so now we have this extensive amount of data. And what our first question is as a researcher, at least in our case, I would say is there behavioral difference? So based on all this information, can I just make a statement, is there a difference between the two treatment groups that I look at, or is there no difference? Is the behavior changed or not? So what we're now trying to do with the BFA is to just kind of come up with one statistical test to say kind of, yes, there is with a high chance, there is a treatment effect or there is not. So what we first do is we compute the average of all these transitions, and then we kind of compute, in this case, it's the Manhattan distance, but in general, it's just important that you know it's a distance between the two groups. So the bigger the distance based on these transitions, the more different, let's say, they are, and the closer or the smaller the distance, the closer they are, and the less different they are. And you can plot this like that. So you have here distance, and you can kind of plot your true group distance that you computed now with this Manhattan distance based on these transitions here. So now you want to, let's say this true distance, you want to compare to what if there is no difference between the two groups. So we need to kind of find a way how to assess the null distribution of if there would be no difference between the two groups. And for that, we just use, in a sense, simple permutation. So we just kind of create a randomized group where we just, I don't know, put together test animals and control animals, and again, compute these Manhattan distance between the average of these transition matrices. And we get, for example, something like this, and then we do this again and again and again, and we do this like a lot of times. So we normally do it like 1,000 times. You can also do more, but just kind of to get a good estimate of how does this distance look like if there is not really a difference between the groups. So you kind of get a feeling of the null distribution. And now you can simply just run a set test comparing your true group distance, so the real grouping, to this kind of null distribution. And you can get a feeling, okay, is there a difference based on the whole behavior profile or all this information we get from the animals using this BFA. So yes, now I want to show you a little bit how this actually looks like applied to real data. This we also, or all experiments we also show in the manuscript. So, and I want to first start with a chronic stress model. Here we have the chronic social instability test, where we have a group of animals that twice per week over a course of 7 weeks kind of got reassigned to different home cages. So this is kind of what then makes the social instability. So they always share cages with different cage mates, while we have the control group down here where they just get handled over the same amount of times, but they stay with the same cage mate. And then 5 weeks after this last shuffling, and they kind of have time to recover, we run a 10-minute open field test. And this first example is more a bit kind of a proof of concept. So now we run the clustering for 25 clusters. We look at, for example, cluster occurrence, and we also look at transition or differences in transitions between these clusters, between the groups. And we can see here, so now this is already adjusted for multiple testing. And you can still see the effect here is quite strong. So we can still, even after multiple testing correction, we see that there are some significant differences in some of the behavior motives. And also here in the transitions, we actually find some differences. And now we just want to see, now we apply the BFA, and that's normally how the plot looks like. And we can see also the BFA detect these differences between the groups. Now to show another chronic stress model that we have. This is a chronic restraint stress model, where we put one group of mice for 90 minutes, for 10 days, for 90 minutes into a restraint tube, while the control mice just get handled. And then after the last restraint test, 45 minutes later, the animals are put into or perform a 10-minute open field test. Also here, we can see, at least when we look at cluster occurrences, that we still, even after multiple testing correction, we find some differences. But for example, in transitions, we don't really find them. Although we see some transitions that are a bit, well, the lines are a bit thicker, so there seems to be some differences, but they just kind of get erased by this multiple testing correction. And now we run the BFA on this kind of experiment, and we also see, okay, there is a treatment effect. So there seems to be a difference between the two groups in their behavior profile. Now I shortly want to show you an acute stress model, just to show you that this doesn't only work with chronic stress, but also with acute stress models. Here we use an acute swim stress. So the mice are put for 6 minutes into like this bucket filled with water to stress them, and then we have the control mice. They just get handled for a little bit. And then 45 minutes later, again, they perform a 10-minute open field test. And here actually now we can really see. So although when you look at the bar plots, you see that there are some differences, maybe not very strong ones, but there are. But due to multiple testing correction, they kind of get erased. And as well here, it's the case for the transitions. However, with the BFA, we're kind of able to still detect these treatment effects. So there seems to be a difference. And this kind of serves for us as a first pass analysis, similar a little bit to an ANOVA, where we can say, okay, so the BFA tells us there seems to be a difference, and now we can kind of go back and look a bit more into what are actually the differences, because that's something the BFA cannot tell us. It cannot tell us what exactly are the differences. It can just tell us, is there any difference? Good. Then I also shortly wanted to show you that this also works for, let's say, more pharmacological interventions. So we had one experiment where we have different injections of yohimbine. That's an alpha-2 adrenergic receptor antagonist, which should lead to a release in norepinephrine, in the brain of these animals. And then we have as controls saline injections. And then we compare this right afterwards again, or we test the animals right afterwards in an open field test. Also here, it's even clear that we see quite strong differences, but again, due to multiple testing correction, also with the transitions, we don't really find anything significant. But if you run the BFA, we do see there seems to be a treatment effect, and that's also what we know from experience. Also, when we look at classical readouts, that there are differences between these groups. So yohimbine has an effect. Then just the last example I want to make for the BFA, just to have a bit of a feeling, kind of what different types of models you can investigate and that it works. So here we have in the locus coeruleus, we have a drug virus injection where we can then actually activate the locus coeruleus and activate noradrenaline release by injecting clozapine. While this is the control groups with just a saline injection, so there should not be a strong release of norepinephrine in the brain. And then again, right afterwards, they were put in an open field test for 10 minutes. Again, just same story. We do see that there are some differences, but again, due to multiple testing correction, they disappear. But with the BFA, we're kind of able to find these differences. Now, I'll show you 5 examples or 5 experiments where the BFA actually showed treatment effects. So, and this almost sounds a bit like, well, it just detects something all the time. And I just want to show you also one example where we didn't expect to find any phenotype and where it shouldn't, like from experience, but also what we then see from clustering, there shouldn't be any difference in behavior. And for that, I want to go back to this acute stress model where we have the acute swim stress. And the control mice, and we not only performed an open field test 45 minutes later, but also 24 hours later. And there we normally don't see any phenotype anymore, and that's also what you can already see here. So here, the differences between the cluster occurrence is quite small. Also here, transitions, of course, there is always some transitions that are different just because there's variance in the data. So you will find differences. But none of them are as significant, and many of them are quite small. And also the BFA here doesn't detect any treatment effect. So after 24 hours, they don't show any difference in behavior anymore, although one of the group has been stressed. Okay, good. So that's kind of what I wanted to say about the behavior flow analysis. As I said, this is just, or our idea was to kind of give or to create a tool that very simply can assess based on this whole behavior profile if there are some differences in treatment or not, with just one readout or one statistical test instance. Now I want to go to the next point, and that would be we also normally want to see a little bit, or what we normally observe, and I think we observe that, but that not every mouse responds in a similar way, for example, to a stressor. So you have animals that respond stronger, others they don't really respond, some even seem like control mice, although they have been stressed. And to compare these kind of profiles now based on all these clusters can be a bit, or it's not so easy to actually do so. So we kind of came up here with this behavior flow likeness score, and I just shortly want to show you again a bit what the issue is. So, so you have an OFT, for example, recording, and now you have this single mice, and you get with the cluster, and you kind of get a behavior profile for all these mice. And as I said, maybe you want to compare between them. You want to see, okay, which animals behave similarly, which don't, and maybe based on this, let's say, treatment response, in a sense, you would maybe also like to see, okay, based actually on this treatment response, can I maybe predict future behavior? So could be again in an open field test, but can also be in a different test where you want to see, okay, how the mice behave here. Does this have any value in predicting what they may be, or how they maybe behave in the future? And as I said, for that, we kind of came up with this behavior flow likeness score, or BFL score, how we call it. And also here shortly, I want to show you a bit what it actually computes to give you a bit of a feeling what we're actually doing. So, sorry. What we have here is, so we have group A, each single point is one mouse, and we have group B, also here, each single point is a mouse. And all of this here now is shown in 2D, of course, because again, we are working with transitions. So we have multi-dimensional space. So this would be actually hard to show, but I'm now showing it here only for 2D to make it a bit simpler, but this can get quite complex in a sense. And what we now do is we take one mouse. This is our sample mouse. And now we want to see, does this mouse behave more like a usual or normal mouse, or usual mouse like belonging to group A, or does it behave more like a mouse that belongs to group B? And how do we assess that? We first of all compute the median centers of this kind of data cloud, let's say, and we also compute it here. So these are the stars that you can see here. And then we just calculate the distance to these median centers. We take these distances and we compute a ratio between them. And that's what we call the BFL score, because it tells us, does it behave more like group A mouse, or does it behave more like a group B mouse? And this now gives us a single readout based on all these transitions that we have from the clustering or the behavior segmentation to kind of estimate a bit the treatment response. So we can plot this like I've done here. This is just an example, and you can see here, for example, these are now the BFL scores for group A, and here we have the BFL score for group B, sorry. And now you can already see a bit. So there are some animals that respond stronger to, for example, a certain treatment if group B would be our stress group, and some respond quite similar to the control mice. And what you can, for example, now also do is you can kind of define a cutoff threshold where you say, okay, these are my non-responder animals, and these are my responder animals, and then you can look at their behavior profiles. How do they behave actually differently? And you can also then try to see, okay, based on this grouping, can I maybe predict how the animal will behave in the future? Will the responder mouse still respond strongly maybe to another stressor, or does this not relate at all? So for the BFL, I again want to show you an example how this can look like when you actually apply it to real data. Here we only have one experiment. We used an IFS, so an inescapable foot shock paradigm, to show this. Here we first had an open field test before the actual foot shock was applied. just to kind of get a baseline behavior. Then at 1 day afterwards, one group of mice got 20 shocks over 25 minutes, and the other group was could just explore the box. Then 24 hours later, we performed another open field test on these animals. Then they went through extinction sessions for 6 days, so field extinction session in the same box, but without the foot shocks. And then on the latest day, that was day 8, again, another open field test, just to, again, assess their behavior or how they behave. Before I now talk about the BFL, just shortly showing you again the BFA. So for OFT1, we see there's no difference between the groups, which is kind of expected because there are not really any groups. They have been treated similarly. And then in OFT2, we actually do see quite a strong difference because the foot shock paradigm is quite a strong stressor. So we do see differences. And then at day 8, so after all this fear extinction, we don't see any difference anymore, at least when it comes to behavior. Okay, so that's the BFA. Now let's go to the BFL. And that's kind of similar to the plot I showed you before. So we now compute the BFL score for each individual animal based on the open field test too. And what you can see here, these are the control animals, and this is already split into 2 groups. So here we have the non-responders. These are all the animals that, based on our BFL score, kind of fall in the same range as the control mice, while here we have the responders, which kind of seem to respond very strongly to the stressor. We also shortly tested again with the BFA if there are actually differences in the behavior profile, which is kind of expected based on how we actually split them here. Of course, there are differences between the non-responders and the responders in their behavior. And what we also tried, and I would say we partially succeeded, to try if we can actually predict freezing behavior during these extinction sessions based on this BFL grouping that we had. And what you can see here is that at extinction day 1, there is not really a difference between the responders and non-responders. Well, tiny, but there's just a trend, I would say. But what we can see is that the non-responders kind of go back faster to baseline, at least a little bit faster compared to the responders. So they seem to show a stronger freezing behavior for longer. Exactly. Good. Yes, and then I also shortly wanted to mention that we are actually not only using the BFL to make this treatment response or response assessment in a sense, but we actually also use it, or you can also use it to compute the effect size. So you can again use these plots, and just by computing the Cohen's d between these and this group here, you can get an assessment of the effect size. And you can then use this effect size, for example, for power calculations, if you want to assess, okay, how many animals do I need for my future experiments? And normally for power calculations, I think what many people use is just one kind of cherry-picked behavior readout, which is normally like one of the classical ones. Again, for an open field test, could be distance moved. And based on that, you do your power calculations. However, here, we kind of assess the whole behavior profile that a mouse shows when it's performing these kind of behavioral tests. So kind of the information that flows into is a bit more extensive. And you can not only do it just for one experiment, but what you can actually also do, you can use these calculations to, for example, compare different numbers of clusters based on their statistical power in a sense. And what you see here for this specific experiment, that 25 cluster, that's the line here, kind of shows the highest power with the least amount of animals. So for this experiment, I would, for example, now choose 25 cluster exactly to hopefully find a treatment effect in the future. And what you can, for example, also do, you can also compare different segmentation tools. Here, as I said, we normally use k-means, but there is, for example, also Bezoid, Vein, and you can also compare kind of these tools based on their statistical power, exactly, using this BFL effect size. Okay, Good. Yes, so that's what I wanted to say about the behavior flow likeness. Again, it's just, again, trying to take all this information that we get from the clustering and make it maybe a little bit more digestible to kind of, with the BFL, get one readout that we can then compare between groups, between individual animals, and maybe even use for the future, for prediction of future behavior. And now we want to come to the last part of today's presentation, where we try to find a way how we, not necessarily quantitatively, but more qualitatively compare behavior phenotypes between different experiments, meaning between different stress models, for example, that we use. And I just really want to show you a bit what the issue could be if you try to do that. So if you say, like, okay, for example, I have a chronic stressor, what we have here. So this is, again, the social instability that I showed you before. And here we have the acute swim stress model. And if I now want to see, okay, either by looking or just by experience, I know they behave kind of different, although they've both been stressed, but chronic stress normally shows a bit of a different behavior phenotype, maybe that then compared to an acute stressor. And when you now look, you can, of course, compare different clusters. You can look like, okay, for example, cluster 1, here it kind of goes down, and here it stays kind of stable. For example, here cluster 14, in chronic stress model, it goes up, but here in the acute stress model also it stays kind of stable. This is, of course, a way how to compare these. And of course, if you really want to know the details of how they differ in their behavior profile, that's the way to go, in a sense. You can also do that for the transitions. However, to just have a nice overview, just see like, okay, can I visualize it somehow that there is a difference how they behave? So yeah, kind of make this a bit easier to digest, also maybe for colleagues who are not such or that are not such experts in the models you use. And here we are now coming up with the behavior flow fingerprinting, or BFF, which in the end is just a 2D embedding based. We normally use UMAP. I don't know if you also maybe heard from TSNE. It's, for example, used, most likely have seen it with, for example, single cell sequencing and so on, where all the single dots are like single cells you have. And you can characterize them a bit. The principle is a bit the same here. So all the different dots here are single mice again. And what you can see now here, we have combined all the experiments I showed you before. So we have the control mice. These are now the control mice from all the different experiments that we have. They're like this gray cloud here. Then we have the chronic stressors, so the chronic social instability and the chronic restraint stress. And you can see that they are kind of, there's not a big overlap between them, but at least they kind of group together at the same corner. So in comparison now to the acute stressors, which are kind of all grouping together up here, there seems to be quite a difference between the behavior that they display. And of course, you can also, for example, see here, normally with Vöikar, we normally see quite a strong response. So you can see that this is really up here, while maybe the acute stress, you find more animals that again behave a little bit more like control animals. So yeah, that's just the kind of a qualitative way to give an overview of how these animals behave in relation to different stress models, for example. Okay, and then I just want to shortly show one application of this BFF, which I think also shows some more stuff quite nicely. So these are now 2 additional experiments. These are actually from collaborators from Rössler in Basel. So these were not performed by ourselves. They had one model where they inject diazepam in 3 different dosages and compare it to a control group where they only inject saline. And the other one was, again, with Vöikar compared to a saline group. And right after the injections, they actually perform 30 minutes of open field test. They also normally have bedding in the open field, and also just in general have a bit of a different setup. So it was also nice to actually show that the whole pipeline in a sense also works on data recorded in a different lab. However, here I just want to show actually only the BFF or the 2D embedding. because here you can actually really nicely see that you have the control groups here, and then you have the diazepam kind of response. Yeah, it's almost a response curve. So different dosages kind of give you a stronger response if you increase the dosage. And the same also is true here for the yohimbine. So you can really nicely see this shift kind of in behavior for the different dosages. Yes, so that's what in principle I wanted to explain and show you today. I think these are the main messages that I think are maybe also helpful for the community, the behavior community, to use. Just a short summary again. So what we propose as kind of a simple tool to assess treatment differences between groups, we have the behavior flow analysis, what we call the BFA, which just gives you one test that tells you, is there a difference in behavior based on the input that you gave me between the two groups. Then we have the behavior flow likeness score, which again gives you kind of one readout, but more in the direction of treatment response, where you can actually compare how strongly do single animals respond to, for example, a certain treatment, and can also be used to compute effect size. And then the last thing I showed you was the behavior flow fingerprinting, so the BFF, where it's just kind of this is more a qualitative tool where you can just compare phenotypes between experiments and compare if they're different or not exactly. And before I come to my last slide, I just shortly wanted to mention one last thing, which I think could be interesting, is that although all this was kind of designed around all this behavior segmentation, clustering, or however you want to call it, tools, however, all the things I showed you down here, I showed you before, in principle can also be applied to the output of a supervised analysis pipeline. So, of course, if you only kind of try to detect one specific behavior, it maybe doesn't make sense because you just have that single behavior that you try to find in your data, and you don't really need to account for multiple testing and all these kind of things. But if you maybe have a model that tries to detect different behaviors, could be in rearing, maybe supported and unsupported rearing, grooming, maybe you even have like subtypes in grooms that you want to detect, or you have a model that actually detects them and then gives you kind of an output that where you can quantify these kind of measures. You can in principle apply all of this also to this kind of output. So as long as you have on a frame-to-frame basis a label that was assigned either by clustering or by supervised model, doesn't really matter for the later analysis in this sense. Okay, good. And yeah, that kind of already brings me to the end of my presentation. Of course, thank you for your attention. I also want to, of course, thank my lab here in Zurich, so the Vootele lab. I also want to thank the Free R Hub here at ETH Zurich that helped us with some of the experiments. And of course, also our collaborators at Roche in Basel, who gave us feedback on the manuscript in general, on the whole pipeline, and also provided us with some data. And yes, maybe just shortly for the people here in the webinar. So with the link, you find the manuscript. You also actually find there a link to a GitHub repository where all this, what I showed you, is implemented in R. So if you're interested to actually apply all these pipelines to your data, this is also available open source, so you can just use it if you want to. So, thank you.